Hello everyone. Today we'll be studying the second topic, that is mitosis and its significance. I am your facilitator, Dr. Girima Nagda, Assistant Professor, Department of Zoology, University College of Science, Mohanlal Sukhadia University, Udaipur. We'll just have a quick recap of what we have studied in our previous video about cell cycle. We can majorly divide the cell cycle into two important phases, that is the interphase and the dividing phase or the M phase. The interphase consists of G1 phase, S phase and G2 phase, where G1 and G2 are the growth phases and S phase is your synthetic phase, and the dividing phase can be mitosis or meiosis. Now we'll be talking about the M phase, which is also known as the maturation phase or the dividing phase, and it is now further divided into two stages. First is karyokinesis, or which is also known as the division of the nucleus, and the second stage is division of the cytoplasm or cytokinesis. Now when we talk about karyokinesis, it can occur either through mitosis or through meiosis, where mitosis occurs in your somatic cells and meiosis occurs in your germ cells. Now when we talk about mitosis, it is the equational division. It is a cell division process. The result of mitosis it results in the formation of two identical daughter cells which are exactly similar to that of the parent cell, each having the same number of chromosomes and similar genetic material as that of the original parent cell. It is called as equational division because the number of chromosomes remains the same, that is, the ploidy is maintained. Mitosis can further be subdivided into the following stages. The first phase is prophase, prometaphase, metaphase, anaphase, and telophase. Now we'll look at all these phases one by one. Now the first is prophase. This is the first phase of mitosis or the first stage of mitosis, and here the chromatin material starts condensing into chromosomes and the nucleus disappears at its end. As you can see in the diagram, we can see the chromatin material is getting condensed and these esters are being formed, which will ultimately result in the formation of spindle fibers during the mitosis. These are the yellow colored structures are your centrioles. So during prophase, the chromatin material condenses itself and it forms the chromosomes. The centrosomes separate and they move to the opposite ends of the nucleus. The centrosomes start to form a framework which is then used to separate the two sister chromatids called the mitotic spindle, which is made up of microtubules, and finally the nucleolus disappears at the end of the prophase and the nuclear envelope disintegrates. The next phase is prometaphase, where we can say that it is the phase just occurring before metaphase and the nuclear membrane breaks down. The kinetochore microtubules invade the nuclear spaces and attaches to kinetochores, and the polar microtubules push against each other, moving the centromeres apart, and you can see the astral microtubules as well as the kinetochore and polar microtubules. During the prometaphase, the nuclear envelope fragments, the fragmentation takes place. Chromosomes now become more and more condensed as compared to the earlier phase. A kinetochore is formed, as we have seen earlier that kinetochore microtubules are also formed. And these are the points where the sister chromatids are attached and the microtubules attached to the kinetochores. The next phase is the metaphase. During the metaphase, the most peculiar thing is the arrangement of the chromosome on an imaginary central equatorial plate, which is known as the metaphase plate. Here you can see the chromosomes are arranged on the central plate. The chromosomes align on an axis called as the metaphase plate, and the spindle consists of microtubules, one microtubule attached to each chromosome at the center, that is the centromere part. Now next comes the anaphase, where the chromosomes break at the centromeres and the sister chromatids start moving towards the opposite poles. As you can see here, the sister chromatids are moving towards the pole. So during anaphase, centromere splits, making the two chromatids free, and then they start moving towards the opposite poles, and the cell begins to elongate, which is caused by the microtubules that are not associated with any kinetochore. The last phase is the telophase, and cytokinesis both occur simultaneously. During the telophase, the chromosomes decondense themselves and they are again turned up into chromatin-like material, thread-like structures. The nucleolus is formed and the nuclear membrane is also formed, and we can see the formation of nucleus. So the nuclear membrane reforms, nucleoli reappear, chromosomes unwind into chromatin, and then as far as cytokinesis is concerned, we'll see in further slides that how cytokinesis occurs in plant cells and animal cells. So during the telophase, nuclear membrane and nucleolus are formed. Short and thick chromosomes begin to elongate to form long and thin chromatin, and formation of cleavage furrow occurs in case of animal cells, and cytokinesis, or finally the division of cytoplasm, take place. So this is all about karyokinesis, where the nucleus has divided, and now we'll be talking about how does the cytoplasm divide itself, or how cytoplasm division takes place, that is cytokinesis. Now there is an interesting thing that this process of cytokinesis occurs differently in an animal cell as well as in a plant cell. In an animal cell, cytokinesis or division of cytoplasm takes place by the formation of cleavage furrow. As you can see here, a cleavage furrow is formed and a contractile ring develops, and ultimately the cells are separated. In case of plant cells, no such cleavage furrow occurs, but a cell plate is formed, which is formed by the Golgi bodies, and this cell plate then separates the nuclei and forms two daughter cells. This is the diagrammatic representation as well as how the interphase, early metaphase, that is prophase, late mitotic prophase, mitotic metaphase, anaphase and telophase appear in cells as well as in diagram. Here you can see these are the different mitotic stages shown in onion root tip. This is your prophase. Here in this cell as well as here in this cell you can see the metaphase, and this cell is depicting the anaphase. As you can see, the sister chromatids are moving towards the poles. So this is a brief about mitosis, where interphase is followed by early prophase, where duplication of DNA has already occurred of DNA and centrioles begin to move into their position. During the middle prophase, spindle fibers attach to the chromosomes, and by late prophase they start lining up on the central plate, and that you can see clearly during the metaphase stage, and the centromes begin pulling chromosomes apart. During the anaphase, the sister chromatids are separated and they start moving towards the pole, and during the telophase, the chromosomes have reached the pole, nuclear material surrounds the chromosomes, nucleolus appears again, and finally after cytokinesis we get two daughter cells. Now the result of mitosis, we have two daughter nuclei, each has the same number of chromosome as the parent cell. If the parent cell is diploid 2n, then the daughter cells will also be diploid, and these two cells are genetically identical to each other as well as the parent cell. The significance of mitosis is it permits growth and repair. In plant, it retains the ability to divide throughout the life of the plant. And in mammals, mitosis plays a very important role As fertilized egg becomes an embryo by the process of mitosis, all your cleavage divisions are mitotic divisions. Embryo becomes fetus by mitotic division, and it allows healing or any kind of broken bone to mend up. Thank you. In the next topic, we'll be studying about meiosis and its significance.